Good morning. At the end of class, I'm going to hand back your exams so you have them to look at. And then if you have any questions about it, we'll go over it then. Okay? Today, we are going to finish protein and protein structure. Okay? And then we're also going to go over nucleic acids and nucleic acid structure. Okay? So that means we will end up being finished with macromolecules. Okay? So at the end, we'll go through. Why is it like a puddle over here?
Someone spilled something in the last class.
Oh, okay. So, careful. So we'll finish that. All right? On Friday, we covered the basic structure of proteins, and we said that proteins are composed of what subunits? Amino acids. Okay? And carbohydrates are composed of what subunits? Monosaccharides is probably the best answer. Okay? We have monosaccharides and polysaccharides are carbohydrates. And those subunits in both carbohydrates and proteins are joined by what? How? Through dehydration synthesis. Okay? And we talked about proteins and joining protein amino acids together to form polypeptides. We also talked about the definition of what a protein is versus a polypeptide. And really that definition depends upon functionality. Okay? A protein assumes that it is a functional molecule doing whatever it does, whether it be an enzyme, a structure, a receptor, a hormone, whatever a protein does. Okay? And the structure of proteins are directly related to their function. Okay? So structure and functional relationship. Now, a functional protein consists of one or more polypeptides. Okay? Twisted, folded, and coiled into a unique shape, and that shape is directly related, directly important for its function. Okay? So here's what some proteins look like when they're functional. Right? You can look at a ribbon molecule, which protein biochemists look at to show different structure. And what do you see here? It's not one long chain of amino acids. It's kind of folded. And if you look at this part right here, there's a little like helix. It's like a little ribbon. Right? And then here it goes back and forth and back and forth. That is a beta sheet. Yes?
How would a ribbon interact with like a receptor, like based on its shape?
So this is just a model to show what it looks like. They take up space like this. So it looks like this. And so you see this groove is important, right? They're trying to show that. So usually on proteins that have functions like this, they'll have an area that is important for an interaction with another protein. In this case, say this groove might bind to DNA. So that the shape of this protein will fit around the DNA in some way and attach to it or bend it in some way. Usually there's some type of area where there's an attachment or interaction. Okay? So, the structure of the protein, which is very important or intimately important for its function, is determined by its sequence of amino acids, which is determined genetically. And we went over this on Friday. Right, we went over this one on Friday. So that last line right here, structure determines function. All right? If you, like Brett said, how does a protein interact with its receptor? All right? It depends upon lock and key. Right? You have to have the right shape for it to work. Now there may be other molecules that fit, right, that have the same key shape, but they don't initiate the function that's there. Okay? Here is an example. You have an antibody on one side, protein from your body, okay, and the protein from the flu virus on the other side. Okay, how many of you have gotten a flu shot yet? Flu vaccine. Get it, or else you're all going to be sick come December. Okay? So here's a protein from a flu virus, which could be the one that's used in your flu vaccine. Okay? All the vaccine is a portion of the virus that they're giving to you to initiate immunity, so you develop the antibodies that you have over there in the orange and blue over there. Okay? That antibody has a portion of it that is the right shape or a similar shape as the protein it is going to interact with. These two areas right here are pretty, what do they look like? Are they close in shape? Yes. So there's going to be interaction there. Now this antibody will bind with this flu virus protein and prevent it from infecting cells. That's how you have immunity for flu virus. Okay? So shape and function relationship, very important. Okay? We said there are 4 levels of protein structure. All right? Primary, secondary, tertiary, and quaternary structure. Okay? Most, if not all, proteins have tertiary structure. Okay? Some proteins will work, but in structure that's in the primary or secondary structure, but most of them have, and their functional part is in the tertiary structure. All right? Primary structure is the sequence of amino acids. Okay, that is determined by genes in your DNA. So the sequence of amino acids, methionine, lysine, proline, tryptophan, right? Amino acid 1, 2, 3, 4, 5, 6, 7, however many it is, that is the primary structure. It'll differ depending upon the protein. Okay? So if I'm going to make a different protein, I'm going to have a different sequence of amino acids. Okay? Secondary structure, all right, is also found in most proteins. It consists of coils and folds in the polypeptide chain. Okay? Secondary structure depends upon hydrogen bonding between R groups. Okay? So hydrogen bonding, and then you have interactions between R groups that allow for tertiary structure. Okay, you'll see this in a second, what it looks like in terms of the molecular models. All right? And tertiary structure is also globular structure. All right, so imagine you have this long chain of amino acids, of beads, right? And now you're going to fold that in certain areas, in certain places. You have an alpha helices or a beta sheet, because it's just stable that way. All right? And then you fold it upon itself again. Okay? It makes it globular. Okay? That's tertiary structure. Quaternary structure is an additional step which depends upon functionality that relies on interactions with more than one polypeptide chain. Okay? So, in other words, there are multiple parts of multiple polypeptides that come together to form a functional unit. Okay? Think of it as a puzzle piece. 4 pieces to a puzzle that make a functional thing. So here's primary structure. All right, here's primary structure of transthyretin. It's an amino acid. It's a protein with X amount of amino acids, what, 125 they say, right? And so This is the structure, glycine, proline, threonine, glycine, glutamine, serine, lysine, blah, 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 glutamic acid, the end. That is your primary structure. Putting the right amino acid in the right sequence. Okay? If I change, let's say this one right here, threonine, and I change that to a tryptophan, what could happen? What's the worst case scenario? I change one amino acid, what's the worst case scenario? I could change the function. Okay? And there's several different ways, but I change structure by replacing an amino acid. I could change function. I might not, the cell might not change function. Okay? And that could happen as well. All right? But the worst case scenario is that for whatever reason, this changes, I end up with a protein that has a non-functional or dysfunctional. Right? Either it's not functional anymore, and doesn't do what it was used to do, or it does something else. Right? In some way, I change the function. Change the shape, change the function. Do we all understand primary structure? Sequence of amino acids. Now, secondary structure. You'll see here, you have an alpha helices and beta pleated sheets. This is still transthyretin. Okay? So it's still the same protein we just saw, the 150 amino acids or so. But there are parts of it where you have hydrogens that are going to interact with each other. What are those dots? Hydrogen bonds. Okay? What you have here is a stable structure. Excuse me. The helices are important because they actually hide hydrophobic or hydrophilic regions. Okay? When you have proteins that span the membrane, you have area within the membrane that is hydrophobic. Okay? So this is most transmembrane proteins that span the membrane have these because they can put non-polar residues on the outside and it doesn't disrupt the membrane. Okay? Beta pleated sheets, again, you have hydrogen bonding and the hydrogen bonds allow for a stable conformation. Okay? This doesn't happen throughout the entire sequence of amino acids. It happens in certain areas. Okay? And that's what you see here. Okay? Here's your tertiary structure. The arrows within that tertiary structure are beta pleated sheets. So it's lots of amino acids going back and forth. And then you have one alpha helices right there. Right here, right? See this? That's a helix. So hopefully you see through this picture that not the whole protein is an alpha helix that's folded onto each other. Okay? It's just an area of it, one part. And different parts of the whole primary structure will have different secondary structure. Okay? But I have these different parts with the secondary structure, and I fold this all over on top of itself. Now I have a burrito, at most. Right? This is kind of what it looks like. It's a globular structure. How many of you been to Moe's or Chipotle? Okay? Do you have had a burrito? Yes? They lay it down. They put the rice, chicken, whatever else, blah, 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 right? Different parts. And now they fold it all up. And it's this globular structure. That's what this is. Okay? Transthyretin. That is the end of structure for one polypeptide. Okay? Is this a functional protein yet? Yes or no? Trick question, we don't know. Why don't we know? Some have a quaternary structure. We don't know if transthyretin functions with another structure. Okay? In this case, it does. Transthyretin functions as a dimer, two parts to it. Okay? So what you see here now are two transthyretin molecules that are laid upon one another. Okay? This is a functional protein. Here's one, here's another one. Okay? That is the functional protein. So now you have multiple polypeptides working together to form a functional unit. Do you understand primary, secondary, tertiary, and quaternary structure of proteins? Good. Okay? Here's some interactions between them. The R groups in tertiary structure can interact. These are examples of those R groups, those R group interactions. You could have hydrogen bonds. Okay? You can have these things called van der Waals interactions. Interaction between the side groups, the methyls, right? They kind of attract, one's positive, one's negative, whatever it might be, right? That's for organic chemistry. You could have covalent bonds that interact between R groups. This is called a disulfide bridge, two sulfurs, and you're putting a bond between them. Okay? And then finally, you could have ionic bonds. If I told you that there's a protein that has lots of cysteines, and they form many disulfide bridges, do you think that protein will be more stable than a protein that does not have cysteines or disulfide bridges?
Yes.
Why?
Because it's connected.
How?
The bridges.
How is it connected? Through the bridge, which is what kind of bond? A covalent bond. Okay? And covalent bonds are the strongest, right? So if a protein has these interactions, it's going to become more stable, right? Because these are going to be more difficult to disrupt. Okay? If a protein only has hydrogen bonds, that might be less stable because they're easier to disrupt. Okay? And the structure determines that. The structure of the sequence of amino acids determines where these interactions occur and how they occur. And they will actually determine stability of that protein. Okay? What do you see here? Give me an example of what type of structure you see here. This is collagen. We assume primary structure. Do you see secondary structure? Where? How? The helix, okay? Do you see quaternary structure? Yes or no? Some say yes, some say no. Do you see quaternary? What do you need to have quaternary structure?
More than one polypeptide chain.
More than one polypeptide chain. Red, green, and blue are each a polypeptide chain. So is this quaternary structure? Yes, it is. Okay? Now, this helix is also assuming tertiary structure that you can't really tell in this diagram. Examples of the importance of this. Hemoglobin. Hemoglobin consists of 4 different subunits. 2 beta globin subunits, 2 alpha globin subunits. Okay? And they're in different colors here. You have 2 in turquoise and 2 in dark blue. You also have heme groups, which are iron that bind oxygen. So 1, 2, 3, 4 subunits that come together to form one functional protein. We all know what hemoglobin does, right? What does it do? I just saw all of you shake your head. Yes, we know what hemoglobin does. Well, tell me, what does it do?
Delivers oxygen to the body.
All right, it binds oxygen to transport it in the body, okay? And that's what you have here. Here's iron is going to bind oxygen within red blood cells, okay, and carry 1, 2, 3, 4 atoms, 4 molecules of oxygen, right, for every 1 molecule of hemoglobin. And red blood cells have lots and lots of hemoglobin molecules inside. So they have a very high capacity to carry oxygen, all right? But that functional unit is important. What happens if we change the structure of hemoglobin? It changes function. And here's an example. How many of you heard of sickle cell disease? Okay? Sickle cell disease is a change in structure, change in primary structure of the protein. One amino acid is changed. Okay? A single amino acid substitution changes the structure of hemoglobin, which then alters its function and its ability to bind oxygen, which causes problems, right, in certain cases. Okay? Just kidding. So here's normal hemoglobin up top. And what it does, all right, subunits, there we go. Molecules don't associate with each other. They bind oxygen, happy, red blood cell characteristic shape. Now, we change glutamic acid or glutamate to a valine by changing one nucleotide in DNA, and now you end up with the different structure within the subunits. The subunits come together, right? Now you have sickle cell, they interact, they clump together, and it causes a sickle cell structure of the cell. Okay? And this is under stressful conditions, right? It can become difficult for those sickle cell red blood cells to move through capillaries, cause clotting, causes spleen damage, could be fatal. Okay? Could be fatal. All right? So, what determines protein structure? The sequence of genes, right? And that's determined by the sequence of amino acids. Okay? Well, but when we change this stuff, when we change certain conditions, salt conditions, pH, temperature, okay, we can disrupt protein structure. How would an alteration in pH alter protein structure? What are you doing when you increase pH? What are you increasing? The concentration of what? Hydrogen ions, H pluses, right? And some proteins are interacting through hydrogen bonds and through ionic bonds. You increase the charge, you increase H pluses around, what's going to happen to the interaction of hydrogen bonds? Is that going to be disrupted? Yes, it will. Would ionic bond association be disrupted? Yes, it will. Okay? So thinking about this, that could lead to a change in shape or loss of shape, which is called denaturation. All right? Cells sometimes are exposed to heat, increased heat, high levels of pH or salt concentrations, and the proteins within the cells are disrupted. Their shape is disrupted. They're denatured. Okay? That denatured protein is biologically inactive. Okay, so if I have an enzyme, I heat it up, I'm going to break it down, it's going to denature it, now it's not going to work as well or at all. Okay? Is this good or bad for cells? Not good, right? Not good. Is it something that cells should have a mechanism to prevent?
Yeah.
Yes. Okay? Because if I break down, so go back to your transthyretin example, if somehow that shape is disrupted, is it more efficient, is it easier for the cell to make a new protein or to correct the misfolding of the original? What's going to take more energy? To remake it, right? It's going to take more energy to remake it. It's going to take more time to remake the protein. So the cells actually have ways of renaturing, of returning a lot of proteins back to their original active shape. Okay? So we have denaturation, right, to renaturation. And some cells do this. Okay? Some proteins can go back on their own. Some can go back with the help of some other cellular machinery. Some proteins do not go back at all. All right, it all depends on the protein we're talking about. Okay? We could predict folding. We can predict interactions between the structure of amino acids. So if we know there's a cysteine here and a cysteine here, at one position in the amino acid and a cysteine at another position, we can predict if they are going to form a disulfide bridge based upon structure. All right? That does not mean that a cell doesn't have other plans for it. We can predict structure, but the cell kind of can disrupt that and have different routes for it to go. Okay? We use X-ray crystallography to determine and predict protein structure. All right? It's we're going to go through it. It's a funky little procedure. Questions on protein structure? All right, so I've been saying that protein structure, the sequence of amino acids, is determined by genes. Genes are units of inheritance, right? They are sequences of DNA. Okay? DNA is a nucleic acid. There are two types of nucleic acids we're going to talk about: deoxyribonucleic acid and ribonucleic acid. Like carbohydrates and amino acid and proteins, nucleic acids are joined and formed by joining monomers into polymers. All nucleic acids are composed of nucleotides. Okay? Nucleotides have a basic structure. Okay? They have a 5-carbon sugar. Okay, that's your 5-carbon sugar. A pentose, right? Off of this first position, that one position right there, they have some type of base, a nitrogenous base. Okay? And then off of this last carbon, they have a phosphate group. Phosphate sugar base. Okay? 1, 2, 3. 3 parts to a nucleotide. Phosphate sugar base. Okay? Two types of nucleic acids: deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. We're going to talk about the differences between the two. Okay? DNA is the genetic material, right? Has genes that code for proteins in the body. Okay? DNA directs the synthesis of RNA, especially mRNA, messenger RNA, right? Which then carries the code for the instructions for building a protein to ribosomes to build the protein, carries out protein synthesis. So, in a nutshell, this is what it looks like. Where's DNA in the cell? Nucleus, and only in the nucleus. It stays there. So, nucleus is covered in eukaryotic cells is contained by a membrane that holds the DNA in, but protein synthesis happens outside of the nucleus. So the message RNA is the intermediate between those. It is coded by the DNA and transmits that code to the ribosome to build a protein. All right? And we'll get more into this when the specifics of this beginning of next semester. But we understand the difference in function between the two. What does DNA do? Has the instructions, codes for genes. What does RNA do? Helps carry out protein synthesis, right? And there's a couple different types of RNA, and we'll talk about it. Okay? Nucleotides. Nucleic acids are really polynucleotides. So we're going to join that nucleotide, that phosphate sugar base molecule we drew, together to form nucleic acids. All right? To form those polynucleotides. How do we join them together? Through what? Dehydration synthesis. Very good. Okay? Important distinction here. A nucleoside versus a nucleotide. A nucleoside is sugar and base. Okay? Sugar and base, no phosphate. A nucleotide has the phosphate. Okay? Questions on the components of a nucleotide? What it looks like. Good? Clear as mud? All right. So here's DNA and RNA. So we just talked about DNA has a different function than RNA. If they have different functions, they probably have different what? Structures. Okay? So here's DNA on the left, double-stranded, and a helix. RNA on the right, single-stranded, right, and does have secondary structure. We'll talk about that, but difference in structure. What do you know? What is present in DNA within the bases that is not present in RNA, and vice versa? T's and U's. DNA has thymine. Right, has the base thymine. Uracil is only found in RNA. Okay, RNA does not contain the base thymine. Okay, so T's and U's are different. Difference in bases probably leads to a difference in structure, difference in function. Okay, so here's another example. Here's the difference. Here's your nucleoside. This right here, sugar base, right? The whole thing is a nucleotide, and they are linked together. Here you see a couple different molecules, 5' N, there's your 5' carbon. This right here, where the base is attached, is your number 1 carbon. Okay, that's going to be important when we talk about DNA replication. So we're going to actually, number them. 1, 2, 3, 4, 5, right? This is your 5' carbon. Your 3' carbon is the important part, right? 5, 3, right? Phosphate. And this ends up giving you, you'll note that goes sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. That's what we call a sugar phosphate backbone to the DNA or RNA strands. Okay? And then the bases are orientated towards the opposite side. Here's your bases. Right? Uracil and RNA, thymine and DNA. The other difference between the two is the sugar. Still a 5-carbon sugar, but in DNA you have deoxyribose, and that has an hydrogen on the 2' sugar, and in ribose in RNA, which has a OH on the 2' in the sugar. Okay? Difference in structure? Yes? Difference in function. Okay? Because we don't want to mix up and add RNA molecules to the DNA strand, right? Yes? No? Agree? We want DNA molecules in the DNA strand and RNA molecules in the RNA strand. This difference in sugar allows that to happen. Okay? OH, oxygen takes up more space, so when the enzymes are making DNA, it's not going to incorporate the RNA nucleotides, only the DNA nucleotides. Okay? The bases, cytosine, thymine, uracil, adenine, and guanine. Right? Thymine is only found in DNA. Uracil is only found in RNA. The rest are shared. Each of them either has one ring or two rings that include nitrogen. That's why we call it a nitrogenous base. Contains nitrogen. Okay, deoxyribose and ribose. This is just going through the other stuff we already went through. All right, but I put it on there in slides so you can have it. Okay. And then the prime, really, which is it's like this, 5 prime. This is the prime. Used to identify the carbon atoms in the ribose, such as the 2 prime, 3 prime. Okay? We went over the sugar phosphate backbone, and adding that sugar phosphate backbone is your dehydration synthesis. That is where we are adding molecules together. We're going to link them together. Okay? So if I had, draw this with me now, we had a 5 carbon sugar, right? Here's your 5 carbon. That's your, draw that again. Here's the carbon. That's your 5 prime carbon. Right, that's your 1. Here's your 3. Okay? Base over here, A, T, C, G, U, whichever one you want, right? And the phosphate's up here. Okay? Down here to this 3 prime OH, we're going to have an OH right, coming off right there, right? To that 3 prime OH, we're going to add the phosphate group. We're going to link the phosphate group to that next one. So here's the next phosphate group. To the carbon and the next nucleotide with the base, right? So this is where, right here, here's another hydrogen off of that, okay? That's where the dehydration synthesis is going to occur. It's going to form what's called a phosphodiester bond. Phosphodiester. Okay? Di refers to two ester bonds, like with the phosphate. Okay, the phosphate attached there. Phosphodiester bond. That's the link between sugar phosphate forming your sugar phosphate backbone. Okay? Okay? And then the linkages among nucleotides and what bases are in order, what nucleotides come in, A, C, T, G, is that sequence eventually determines the sequence of amino acids and determines the structure of the gene that you're in. Okay? RNA molecules, we just went over structure of these. DNA is how many strands? 2. In what structure? A double helix. And RNA is how many strands? 1. And has some secondary structure, but it's not. Right? When we go through the structure of DNA, and RNA, when we talk about replication, we'll get into this more. DNA is what we call anti-parallel. 5' to 3'. Important that I told you understand the numbering of the carbons. 5' to 3', you're always going in that direction. 5' up top, 3', 5', 3', 5', 3' from top to bottom. Well, the other strand is going to go in the opposite direction. Think about as a two-way street separated by double yellow lines. Okay? Oncoming traffic is always going in opposite directions, right? So 5' to 3', top to bottom, what's the other one going to go from top to bottom? 3', 5', 3', 5'. It's going in the opposite direction. Okay? The bases are also paired in a certain way. A to T in DNA, C to G in DNA. Why am I not talking about base pairing in RNA? Because it's a single-stranded molecule, right? There is some secondary structure where A would go pair with U, C would still pair with G, but that is not as important as it is in DNA. A to T, C to G, okay? This is what it looks like, okay? You have structure. Here's your 5' to 3', right? Traffic, let's go 5' to 3' going this way, right? 5' to 3' going that way. Two-way traffic, right? Here's your base pairing, and they link together by what type of bonds? Hydrogen bonds hold that together. I told you that some secondary structure is present. Some base pairing is present in RNA. Here's an example. Transfer RNA, which is important in protein synthesis. That structure is held together by interactions among the single strand of RNA, right, to form that secondary structure. And you'll see that actually there's pieces of this that point out. Those are important when we talk about transcription translation. All right? But A to U, C to G. Okay? And these DNA and RNA can be used for determining evolution. How we can look at the sequences of DNA and tell how closely or far apart organisms are related. So we can also look at the structure of RNA and determine that as well. All right? So it gives you an idea of how closely two organisms are related. And this, the evolution of molecular techniques and sequencing, determined that archaea, that whole domain, is more closely related to eukarya than bacteria. That's all based upon this, clues in evolution. Okay? Questions on DNA, RNA structure, protein structure, and the importance of both? No? Yes? Maybe? Okay.